We will post new science and technology updates here. New U.S. population study projects steep rise in cardiovascular diseases by 2060. By the year 2060, projected rates of cardiovascular risk factors and disease will increase significantly in the United States, according to a new study. More you might like a major step forward for organ biofabrication. Heart disease, the leading cause of death in the U.S., is so deadly in part because the heart, unlike other organs, cannot repair itself after injury. That is why tissue engineering, ultimately including the wholesale fabrication of an entire human heart for transplant, is so important for the future of cardiac medicine. To build a human heart from the ground up, researchers need to replicate the unique structures that make up the heart. This includes recreating helical geometries, which create a twisting motion as the heart beats. It's been long theorized that this twisting motion is critical for pumping blood at high volumes, but proving that has been difficult, in part because creating hearts with different geometries and alignments has been challenging. This advancement was made possible using a new method of additive textile manufacturing, focused rotary jet spinning, FRJS, which enabled the high-throughput fabrication of helically aligned fibers with diameters ranging from several micrometers to hundreds of nanometers. Developed at SEAS by Kit Parker's Disease Biophysics Group, FRJS fibers direct cell alignment, allowing for the formation of controlled tissue-engineered structures. The research is published in Science. This work is a major step forward for organ biofabrication and brings us closer to our ultimate goal of building a human heart for transplant, said Parker, the Tarr Family Professor of Bioengineering and Applied Physics at SEAS and senior author of the paper. This work has its roots in a centuries-old mystery. Over the next 3 centuries, physicians and scientists have built a more comprehensive understanding of the heart's structure, but the purpose of those spiraling muscles has remained frustratingly hard to study. Our goal was to build a model where we could test Salin's hypothesis and study the relative importance of the heart's helical structure, said John Zimmerman, a postdoctoral fellow at SEAS and co-first author of the paper. The first step of FRJS works like a cotton candy machine. A liquid polymer solution is loaded into a reservoir and pushed out through a tiny opening by centrifugal force as the device spins. As the solution leaves the reservoir, the solvent evaporates, and the polymers solidify to form fibers. Then, a focused airstream controls the orientation of the fiber as they are deposited on a collector. The team found that by angling and rotating the collector, the fibers in the stream would align and twist around the collector as it spun, mimicking the helical structure of heart muscles. The alignment of the fibers can be tuned by changing the angle of the collector. The human heart actually has multiple layers of helically aligned muscles with different angles of alignment, said Hubin Chong, a postdoctoral fellow at SEAS and co-first author of the paper. This is important when it comes to building a heart from scratch. Take collagen for instance, an extracellular matrix protein in the heart, which is also a single micron in diameter. It would take more than 100 years to 3D print every bit of collagen in the human heart at this resolution. FRJS can do it in a single day. After spinning, the ventricles were seeded with rat cardiomyocyte or human stem cell-derived cardiomyocyte cells. Within about a week, several thin layers of beating tissue covered the scaffold, with the cells following the alignment of the fibers beneath. The beating ventricles mimicked the same twisting or ringing motion present in human hearts. The researchers compared the ventricle deformation, speed of electrical signaling and ejection fraction between ventricles made from helical aligned fibers and those made from circumferentially aligned fibers. They found on every front, the helically aligned tissue outperformed the circumferentially aligned tissue. Thanks for watching. Please subscribe my channel for more videos.